Assalamu alaikum. My name is Zunaira Zahid. My roll number is 021810. Hope you're all safe and sound in your homes. Today I'm gonna present a very interesting topic, which is hemoglobin. Now, what is hemoglobin and where it is found? Hemoglobin is actually an iron containing oxygen transport metalloprotein in the RBCs of almost all vertebrates and some invertebrates. Hemoglobin is a red pigment that is present in the RBCs of blood, and its composition, it is made up of iron and protein, and its molecular weight, which is 68,000, means it's a very huge molecule. Now come towards the introduction. The main function of red blood cell is to transfer O2 from lungs to tissues and simultaneously transfer of CO2 from tissues to lungs. So to accomplish this function or complete this function, RBCs has hemoglobin, which is represented by HB. HB is a globular protein and it is made up of iron and protein. As you can see in the name, heme means iron and globin means protein. Each red blood cell has 640 million molecules of HB, means one RBC has 640 million molecules of HB. Hemoglobin protein constitute 1 to 3% of the red blood cell. In red blood cells, 1 to 3% is protein and the other molecule is heme group. Hemoglobin is the iron containing oxygen transport melatoprotein in red blood cells of all vertebrates, and the structure of hemoglobin was introduced or elucidated by Max Proust. Max Proust is also known as the father of X-ray crystallographic method. There are many types of hemoglobin: HbA, HbB. Now HbA is the most common. HbA is hemoglobin that is found exclusively in RBCs of adult, and it is composed of 4 polypeptide chains. The structure of hemoglobin is a terameric protein with quaternary structure. Terameric means two sets of similar units, that is alpha 2 and beta 2. Hemoglobin, the word is derived from two words: heme mean iron and globin mean protein. Heme is the prosthetic group that mediates reversible binding of oxygen by hemoglobin. Heme helps mean heme क्या करता है, help करता है hemoglobin को for binding of oxygen, and globin is the protein that surrounds and protects the heme molecule. You can see the structure of hemoglobin here, that is iron in the center in red color, and there are 4 porphyrin ring, as you can see here, and that is protein portion that is in blue color, and there are certain bonds to which it is connected, and we will discuss it later. Heme. फर्स्ट ऑफ ऑल वी हैव टू स्टडी अबाउट हीम. व्हाट इज़ हीम? हीम के पास 2 पार्ट्स होते हैं, एक ऑर्गेनिक है और दूसरा इनऑर्गेनिक है. ऑर्गेनिक पार्ट इज़ द प्रोटोपोरिफाइरिन. You can see these rings, 4 rings, that is organic part, and in the center, which is in red color, that is inorganic part, and it is called iron, and it is also a metal. 4 pyrrole groups, that is A, B, C and D, linked by methane bridges, linked by methane 1, 2, 3, 4 methane bridges. Ferrous in heme act as a prosthetic group. Ferrous mean iron that is in the center, that acts as a prosthetic group, and ferrous ion resides at the center of tetrapyrrole ring. Tetra means 4. There are 4 pyrrole rings, so we can say tetrapyrrole ring. It is also called hydrophobic heme pocket. There is a space between porphyrin and heme group, which is called heme pocket. Hemoglobin has 4 heme, so it is evident that hemoglobin can carry 4 oxygen atom at one time. स्ट्रक्चर को अगर हम और ज़्यादा देखें तो हमें ये पता चलता है कि आयरन अटैच्ड होता है टू ईच पायरोल रिंग, एंड देयर आर 4 पायरोल रिंग्स, एंड 4 ब्रिजेस हम देखते हैं कि कहाँ कहाँ पे कौन कौन से साइड चेन्स अटैच हैं. तो साइड चेन्स आर 8, so 4 is methyl chains and CH2, which is carbonyl. There are you can see here there are two vinyl chains, and there are two propionic acid CH2CH2COOH. here is its name, propionyl भी कहते हैं इसको, और there is two vinyl and there are four methyl chains. Now, iron is bound strongly to the globular protein. आयरन प्रोटीन के साथ किस तरह से अटैच होती है? वाया एमिडाजोल रिंग. वो जो कोवलेंटली अटैच होता है आयरन और प्रोटीन, उसको हम आइमिडाजोल रिंग कहते हैं. ऑफ F8 histidine residue, also known as the proximal histidine below the porphyrin ring. Here in blue color is the protein portion and upper portion is iron portion. Now this iron and this protein are connected covalently through a amidazole ring, which is a histidine protein. A sixth position can reversibly bind oxygen by a coordinate covalent bond completing the octahedral group of 6 ligands. Now in the structure there are 6 ligands, and sixth position can reversibly bind oxygen by a coordinate covalent bond. That is the sixth position, and it bind oxygen bond with this, and one oxygen atom is out of the plane. You can see oxygen binds in an end-on bend geometry where one oxygen atom binds Fe and the other protrudes at an angle. It means that iron is connected to one iron. Oxygen is connected to to central ferrous atom or iron through a coordinate covalent bond, and one oxygen atom is protrudes out of the angle. And when oxygen is not bound, a very weakly bounded water molecule fills the site, forming a distorted octahedron. Means when oxygen is not bind and the hemoglobin is oxyhemoglobin, then a water molecule takes its place and distortion of geometry takes place. Next we have to study further. You can see a molecule of 4,2 acts as 6 ligand. There the oxygen molecule act as a sixth ligand, and porphyrin ring in heme with its particular arrangement of 4 methyl 2 propionates and 2 vinyl substituents. As we can see earlier कि यहाँ पे 4 methyl 2 propionate and 2 vinyl substituents हैं. Then in the oxy form, the ferrous atoms sit out of the plane porphyrin by about 0.6 angstrom. When hemoglobin has no oxygen, and means when it is not saturated, then oxygen is not present obviously. So what happens at this stage? In the oxy form, the ferrous atoms sit out of plane of porphyrin. That ferrous atom is out of plane by porphyrin due to the unavailability of oxygen. And when O2 binds, and because deoxyform ferrous atom sit out of the plane porphyrin by atom 0.6 angstrom. इस तरह से क्यों होता है? Because steric repulsion between histidine F8 and porphyrin plane. That is histidine and that is porphyrin plane. इन दोनों में रिपल्जन होती है, जिसकी वजह से जब ऑक्सीजन बाउंड नहीं होता है तो Fe एटम जो होता है वो पोरिफाइरिन प्लेन से आउट ऑफ प्लेन हो जाता है थोड़ा सा, और डिस्टॉर्शन हो जाती है स्ट्रक्चर में. एंड व्हेन O2 binds, the steric repulsion is minimum. When O2 binds, the steric repulsion is minimized and ferrous atom comes to the plane. Oxygenation changes electrostatic state of Fe2 of heme. The oxidation state of central Fe is +2, and after oxygenation it changes. Now you can see the molecular structure to further enhance your learning. There are a red color heme group, and there are 4 alpha, 2 alpha chains and 2 beta chains. Mean there are 4 amino acid chains, protein chains. Iron containing pigment called heme attached with protein globin. Heme is iron porphyrin complex called iron. Globin is the protein, as I can explain earlier. Now we have to study about the mechanism of oxygen binding, how oxygen binds to hemoglobin. So, in the tetrameric form of normal adult hemoglobin, the binding of oxygen is a cooperative process. The binding affinity of hemoglobin for oxygen is increased by the oxygen saturation of the molecule. Oxygen saturation mean amount of oxygen in blood is more, that is 95 to 100% oxygen. The binding affinity of hemoglobin for oxygen is increased by the oxygen saturation of the molecule, with the first oxygen bonds influencing the shape of the binding sites for the next oxygen in a favorable way for binding. It means that when one oxygen is attached to one heme group of the hemoglobin, then it become easier for the other oxygen atoms to attach to these heme groups. This positive cooperative binding is achieved to steric conformational changes. Uh, Now, what is oxygenation? Oxygen binding to hemoglobin is due to the effect of the ligand binding site of one heme group on the ligand binding affinity of another heme group. It means that too far apart to interact means जितने दूर मॉलिक्यूल होंगे उतने ज़्यादा इंटरेक्ट करेंगे 25 to 37 एंगस्टन अपार्ट. अगर इतने ज़्यादा दूर होंगे तो लिगेंड बाइंडिंग affinity होगी so that it can attach oxygen can attach to heme group. Mechanically transmitted between heme groups by motions of the proteins, this means that the molecule changes shape. Now there is a mechanism, that is Proust's mechanism, in which Hb has two conformational state. One is T state and the other is R state. T state means T state is also called deoxy state and R state is also called oxy state. In deoxyhemoglobin, quaternary interactions are constrained, means that hemoglobin is in the T state, that is 10 state. When O2 binds in T state, oxygen is not bind, and in R state O2 binds and it relaxes the quaternary structure, which is called relaxed state. Now, when O2 binds, oxygen pulls some of the electron density from ferrous atom. Ferrous atom is in the center. You can see the structure. Oxygen pulls some of electron density from ferrous atom. As a result, electron density in the iron atom reduces and it can move more to the center of plane of protoporphyrin. Now oxygen pulls iron atom. The porphyrin structure forms alpha helix. So when binding takes place, there is a surface to surface interaction between adjacent polypeptide chains. This interaction forces the plane of the ring sideways towards the outside of the tetramer and also induces a strain in the protein helix containing the histidine as it moves nearer to the iron atom. जब भी इंट्रैक्शन होती है तो टेट्रामर स्ट्रक्चर है हमारे पास, तो इट इंड्यूसेस अ स्ट्रेन इन द प्रोटीन हाइलेक्स में एक स्ट्रेन आ जाएगा और हिस्टडीन होगा वो आयरन एटम के पास आ जाएगा. This strain is transmitted to the remaining 3 monomers in the tetramer, where it induces a similar conformational change in the other heme group sites such that binding of oxygen to these sites become easier. और बाकी heme group के साथ भी इसी तरह से होगा और दूसरे heme groups का यहाँ पे attach होना ज़्यादा easier बन जाएगा. You can see the oxyhemoglobin and deoxyhemoglobin. When oxygen is attached, structure is changed, and when oxygen is not attached, structure is totally different. Deoxyhemoglobin has an extensive network of salt bridges which stabilize T state. Deoxyhemoglobin के पास T state होती है. Deoxyhemoglobin stabilize करता T state को. These ion pairs are torn in transition from T state to R state. This is T state, which is this is R state, which is oxyhemoglobin, and it is T state, that is deoxyhemoglobin. Now we have to study the oxygen hemoglobin dissociation curve. In this curve, the portion of hemoglobin in its saturated form on the vertical axis, we plot a graph between hemoglobin in its saturated form on the vertical axis and oxygen tension on the horizontal axis. The graph comes in a sigmoidal shape. Sigmoidal means ellipsoidal, something in the form of ellipsoidal. Uh, Sigmoidal shape of HB dissociation curve shows characteristics of cooperative process. Cooperative process means oxygen binding process. Now right to left dissociation हो रही है in lungs. In lungs, O2 tension is high, means 100 mmHg. It means it provides complete saturation. Obviously, in oxygen, oxygen is very huge in amount, and it provides complete saturation of hemoglobin molecule in lungs. And when hemoglobin molecule release O2, it is a gradual process. When blood leaves the lung for peripheral tissue, AHB releases its load and percentage of oxyhemoglobin decreases, and oxyhemoglobin become decrease in amount. Now left to right in the graph, you can see left to right in the graph, you can see by there takes place binding of oxygen. Okay, there takes place binding of oxygen. A first molecule bound causes structural changes that influence the binding of next molecule. When one molecule of oxygen is attached to one heme group of hemoglobin, then structural changes takes place, and these structural changes influence the binding of next oxygen molecule to heme group. Initial slope is low because there are so much hemoglobin, and it become competitive for the first O2. Once oxygen molecule is bound to one HB's subunits, it increases the O2 binding affinity of its other subunits, thereby and so increasing slope. Middle portion has increased slope. Hope this topic is clear. Thank you for listening.